Welcome back to the cell signaling course. We'll start from where we left in the last class, and if you recollect, we were discussing different types of ion channels as receptor. And one of the beauty of such ion channels was that they were conducting the signaling process. They were basically serving as a receptor which is doing the signal transduction as well by allowing the passage of ions through them. One of the channels which is very unique in its property and is found in many body parts is water channels, and we will discuss how another type of receptor which is in the class of nuclear receptor, they mediate the signaling. So let's start with the channels which have the property of conducting molecules. We have seen examples of ion channels as of now. Now we'll see the example of these channels which have the property of transducing water molecule and thus ensuring the signaling goes in the right direction. So what happens here is that we noticed in the previous class that ion channels of different types, they had a close conformation when ligand voltage difference or pressure, something like that was applied, and in some cases the leaky channels, they acquired a conformation which was more of an open type, allowing the ion flow. We noticed in that case, some cases the channel was opening and allowing ion flow inward, in some cases channel were opening and allowing outflow. Largely it was potassium which was outflowing, and sodium, calcium were something which were moving inside. We will be discussing a particular type of channels which are called water channels, and they are also called aquaporins. Basically the pores which allow the passage of water molecule. So here also you will have these channels, they will be allowing the passage of the water molecule. Just remember, these are not like ligand gated channels or voltage gated channels, rather they have some control in this region inside the interior surface of the channel which is in the membrane where water molecule is passing. So there are specific residues which are aligned over there which allow the charged water molecule to pass through this passage where largely you will have the lipid hydrophobic environment. So for discovery of these aquaporins, the channel which allow water movement, Peter Agre was given Nobel Prize in chemistry in 2003. In previous class we noticed that how discovery of TRPV channels fetched Nobel Prize in 2021 in physiology or medicine to Julius and Patapushian. So it seems that the signaling world and the associated discoveries have attracted so many Nobel Prizes, so it's very important to understand them. So in this case of aquaporin, these are largely the plasma membrane bound channel. So a good percentage, or if I should say that if there are 10 different types of aquaporins, 80% of them or 70% of them are more localized to the plasma membrane where they mediate the movement of water primarily and in some cases glycerol also. Moreover, some of these forms of the aquaporin, they are present in the ER membrane and they ensure that the ER homeostasis is maintained. You can understand that ER, if you think about a cell and you think about the ER membrane present all over the cell, the cytosol and the material inside the ER, their concentration, osmolarity need to be maintained. So you will need channels which allow the movement of water molecule like this. And the most prominent type of water channels, they are present at the cell surface allowing the movement of molecules. So where do these aquaporins play very important role? We all understand the process of excretion And as soon as we think about process of excretion, always kidneys come into our mind. However, let me explain this, that skin is also one of the excretory organ. The sweat which is produced, that is serving as a excretory product secretion entity. So we know that in kidney, nephrons, they are the one which actually maintain the level of water circulating in our system as part of the blood, which may have high osmolarity versus low osmolarity. When you have high osmolarity, the blood volume is reduced or water content is less. So normally what happens in the kidney, the blood which comes from systemic circulation, here in the glomerulus, it is filtered, and largely water is filtered here, and small molecules like glucose, amino acids, waste products like urea, they are secreted. They are filtered over here. Now the job of this thing is to take the waste product into the ureter, but most of these molecules which have gone out, large amount of water and glucose and amino acid, they will be absorbed back. In blood circulation. We understand that there are blood vessels around it which are waiting for these molecules to now move out of the nephron and be taken back into the blood circulation. Right. So one of the biggest thing which is being taken back in volume wise and concentration wise is water molecule, and this is mediated by the two types of aquaporin, that is called aquaporin 1 and the other one is aquaporin 2. If I write it here as aqua 2. So bulk of water which is being reabsorbed through kidney and particularly at the nephron level is by aquaporin 1. So almost 80% to 90% of, or more than 90% of water which is being filtered here is reabsorbed back in the early part of the nephron, and that is mediated by aquaporin 1. The later part of the nephron here, some extra water can be retained depending on the environmental condition. If there is a dehydration situation, then there will be more water which will be taken back into the system, and that is where the aquaporin 2 comes into picture. While this aquaporin serves allowing, or while this aquaporin 1 allows the bulk of movement of water and does not require any signal, it is always present on the surface of these cells which are here, but the aquaporin 2 needs a signal to be present on the cell surface and allow the more water molecule to be taken back into the system. Now let's talk about the other type of channels or receptors. I would not say channels because these are not necessarily present on the cell surface. Rather, I would say these are the ones which are soluble type of receptor. So they will definitely be not present on the cell surface. When I say soluble, it just means that they are present in not bound to the membrane, and they could be present in cytosol or they could be present in the nucleus. The binding of the signal takes this receptor to the nucleus. Whether it is, if it is present in the cytosol, it takes it to the nucleus. If it is present in the nucleus, it activates the receptor. In both scenario, end result is the transcription. Most of the steroid hormones which we know, or the hormones which are not steroid in origin but do not bind to the receptor on the cell surface, rather they have some sort of transporter on the membrane, and that allows the entry of the hormone. Such a hormone should find a receptor either in cytosol, or the receptor is present in the nucleus, bound to DNA elements but inactive. The movement of this hormone or the bioactive molecule allows this to be active and transcription is followed. So in both scenario, you need this complex of the receptor and ligand. If I say that ligand could be hormone, this could be a small molecule, and so on. So you have these hydrophobic signal or hormones which generally work through these receptors. So let's take a scenario of this hormone which is called ecdysone. This is a hormone which is a primary insect hormone which is required for developmental transition and metamorphosis. What exactly does this mean? That many of the insects undergo during their developmental process, they form larvae of different types. You can name them as larva 1, larva 2 and larva 3. This is the most mature form of the larva. And then this larva turns to pupa, and pupa gives rise to adult. So this ecdysone hormone is important for change from one stage of larva to the other stage of larva. This is what is called developmental transition. And then when it moves to pupa, this is like metamorphosis, because the larva looks very different from how pupa looks, and from pupa the adult looks very different. So you will need surge of this hormone ecdysone at different time points to ensure that proper development of the organism happens. So this hormone is synthesized in some other glands in these organisms, but they need to be carried to the target cell. You remember this concept of target cell, because in the signaling, the target cell is the one which is doing the signaling. Now here, when they are delivered in the vicinity of the cell, they enter inside the cell cytosol. And since these are steroid type of hormone, they freely move inside. In particularly case of ecdysone receptor, the receptors are present in the cytosol. And these are also heterodimeric type of receptor. One is called ecdysone receptor, the other one is called ultraspiracle. This heterodimeric combination of the receptor binds to this ligand which is called ecdysone. If I just write EC. Remember that some of these steroid type of hormones or ligands, they exist in a precursor condition and active condition. So in case of ecdysone, there is a molecule which will be ecdysone, which will be converted into 20-hydroxy ecdysone, and this is actually the active molecule. So when this active molecule is generated in the cytosol, it binds to the receptor, heterodimeric receptor, and this heterodimeric receptor goes inside the nucleus, binds to these DNA elements, and then allows the transcription. Transcription of multiple things. Transcription could be the gene which will allow the changes, transcription of the genes which will make more ecdysone receptor, or transcription of something which ensures that that larva is now ready to pupate, or pupa is now ready to undergo this metamorphic change and emerge as an adult. So this is what happens, that example shown here, that this is your larva and this is your pupa. This is ecdysone receptor which is present in the cytosol, and these are ecdysone molecule. It's written 20E, meaning this is the active form of the ecdysone, and these arrows suggest that level is high. When this transition is required for metamorphosis, you will need more levels of ecdysone. Once this ecdysone enters inside the cell, it binds to the receptor. This receptor now moves inside the nucleus, where it binds to the DNA and carries out transcription. What you would have noticed by now that there are certain regions in the DNA, they are just upstream of where your gene of interest is present. And some of these regions are regulatory regions where you have promoter sequences. So all different cases where you saw a molecule is entering inside, it has transcription factor activity. So all these ecdysone receptor which we are talking about, they should have a transcription factor activity, which means that they should first bind to the region of the DNA, the promoter region. For binding, they should recognize a specific sequence, and then they should be able to activate RNA polymerase for transcription. So this minimum ability of these receptors to bind to the DNA, and that to very specific sequence, makes them very specific to one type of expression of target genes. So all the target genes which needs to be downstream of this ecdysone signaling, they should have this common element in their promoter region, so that your ecdysone receptor as a heterodimer with ecdysone receptor and USP should be able to bind and activate the transcription. This is the basic minimum requirement. The other receptor which works in a very similar way is androgen receptor, which actually responds to the hormone which is called testosterone. We know that male have more of testosterone, and this is also a steroid type of hormone. So these receptors which bind to steroid hormone testosterone, they are called androgen receptor, and they are also in the family of nuclear receptors. So there are multiple different families of nuclear receptor. There are 7 different families of nuclear receptors, and there are 50 odd such receptors are present. Among them, one of them is in the family of androgen receptor. So here also this androgen receptor can bind to entity which is testosterone, or the active form of this which is called dihydrotestosterone. Very much like in the previous case, you saw the ecdysone or 20-hydroxy-ecdysone or 20E. Here also the ligand is testosterone or its active form. So this testosterone which is delivered to the cell, it permeates inside, binds to its receptor which is androgen receptor here. This androgen receptor will go inside the nucleus, allow the transcription. Same thing which I explained to you in the previous slide. Here also this region which is promoter has specific DNA elements which allows this human androgen receptors binding. It is needless to mention here that all these receptors which we are talking, they will also have some specific domain arrangement. One domain which allows the binding of the ligand, another domain which allows the transcription. So at least DNA binding ability, at least ligand binding ability, and then activating the polymerase for the transcription. So some of the domains which are present are called activation function domain in these receptors. They are called activation function domain in addition to DNA binding domain and ligand binding domains. In case of example of such receptors which are nuclear type receptor present in cytosol, present in nucleus. You have another receptor which is called thyroxine receptor. But broadly, this will be in the class of nuclear receptors. So unlike what we saw in the previous two cases where the receptor was present in cytosol, here receptor is not present in the cytosol. What happens here, that T3 hormone uses this transporter and gains an entry inside the cell cytosol. This molecule is very small, so it directly enters the nucleus. So the receptor for T3, the thyroxine hormone, is present inside the nucleus, but it is bound by a repressor. This repressor is not allowing the transcription to happen, the scenario which is shown here. But when the T3 comes, it displaces the repressor, binds over there and now puts the activator. This is your repressor, as I told you. So this complex is unable to do the transcription, but here this complex of T3 activator and the T3 hormone receptor, which is called THR, thyroxine hormone receptor, is capable of doing the transcription. So these receptors which are present over there, they are of two different types, the alpha type, 3 different types of alpha and 3 different types of beta, and they form homo or heterodimeric situation. So the receptor which you see inside the nucleus bound to the DNA element is dimeric one. This seems to be a common theme among different receptors which we have studied, that receptor dimerization minimum happening to ensure that activation of this receptor goes on. Now, what will happen to these receptors? As I said, they are dimeric, they bind and activate transcription, and particularly the receptor in alpha family, receptor 2 and receptor 3, although they can dimerize, but they cannot bind the ligand, which is T3 here. So in some sense, if they cannot bind to T3, they cannot activate the transcription. And very much like what I told you in previous 2 slides, this region of the promoter should have a specific DNA element which ensures the THR binding. Now what happens to the function of these THRs and different organs which is being affected? You know that thyroid has very important function in metabolism, and energy aspects of the organism. So there are multiple thyroid functions which are affected. Let's see the set of function which is affected by THR beta type of receptor. If there is a problem in the THR function, it will give a situation like goiter in the thyroid. First of all, it will disturb the axis which is seen in thyroid, pituitary and hypothalamus. So there are signals like this and then feedback signal onto pituitary and thyroid. So maybe if I draw it much clearer here, hypothalamus is placed above, then you have pituitary, and then you have thyroid. So normally hypothalamus signals to the pituitary, pituitary signals to the thyroid for production of T3 and T4 hormone. When there is enough of signal, it will send the feedback, negative feedback, which should stop the T3 T4 synthesis signal. And this, since it is affecting the metabolism, all different tissues in the body, they are regulated or affected by thyroid. So it affects the thyroid itself, where goiter-like situation can come. In the ear, the perception, sensory perception of hearing, that is affected because cochlear development can be affected by the thyroid malfunctioning. The vision process is affected by thyroid malfunctioning. Heart function, particularly a situation called tachycardia, arises when THR beta family of these nuclear receptor for thyroxine are not working fine, and nervous system, particularly lot of brain region development is affected. So the functions which are mediated by THR beta are more widespread for the fact that all three forms of THR beta, whether it is 1, 2 or 3, all can bind to T3, thus they can form that active complex with THR and the activator and the T3. So they will have more widespread effect than THR alpha. So what we see here is that THR alpha, that has effects on bone density and bone health. It again affects the heart, but here the situation which arises from the T3 malfunction or THR malfunction is called brachycardia. There are digestive system related issue which is in the family of or in the condition of digestion related, indigestion and so on, and then again it affects the nervous system development. Maybe some specific brain areas are affected. So what you notice is that these receptors, which are in a very different class of receptors, basically I classify that as a soluble receptor, or what is known about them to be a nuclear receptor, that is getting affected by the presence or absence of the ligand. Also there could be some mutations which will be causing these effects, and these are very unique type of receptors because they are mediating the signal transduction by themselves. So what we have learnt in today's class is that aquaporins, which are managing the water content in the body by allowing the cellular uptake of that, and recent observations suggest that they are also involved in cell migration, which basically suggests that the leading edge in the direction where cell is moving, you have a lot of aquaporin of specific type located. The cytosolic or nuclear receptors, they bind to ligand and then activate transcription. These ligands could be present in cytosol and are inactive in the absence of ligand, or they could be present in nucleus. When they are present in nucleus, they are bound to chromatin, but they are also bound with the repressor, thus they are inactive. So the presence of ligand activates the entire complex. Insect metamorphosis, particularly the larval development, is mediated by these ecdysone receptor, which we saw was present in cytosol in a heterodimeric combination. Binding of ecdysone took it inside the nucleus. We also noticed that the active form of the hormone which was important for this process. The androgen receptors, which bind to testosterone or dihydrotestosterone, they generate the response relevant to the downstream of testosterone or androgen function. And we realize that thyroxine hormone T3, which binds to the receptor, but this time the receptor is not in the cytosol, rather it is present inside the nucleus, bound with repressor and thus inactive in terms of transcription. When T3 binds, it activates the transcription, which helps in the mediation of metabolic homeostasis. Thank you.